Point mutations are mutations that substitute one nucleotide base for another. Transition mutations are when a purine base is exchanged for another purine, or a pyrimidine base is exchanged with another pyrimidine. Transversion mutations are when a purine base is exchanged for a pyrimidine or vice versa. And just for a quick review, purines are the bases A and G, while pyrimidine bases are C, T, and U. Point mutations can have 4 different effects at the protein level, which are missense, read-through, silent, and nonsense. Missense mutations are mutations where the codon is changed and now codes for a different amino acid. Read-through is when a stop codon is replaced with a new codon that codes for an amino acid. Silent mutations are when a base is changed but the base still codes for the same amino acid as before. And lastly, nonsense mutations change the existing codon into a stop codon. Here we are given a coding strand of DNA, and we will use complementary base pairing to find the template strand. To do this, A will become T, G will become C, C will become G, and T will now be A. Transcription will occur to give us our mRNA sequence. Translation then happens to give us our proteins. Using the codon dictionary, we can figure out what codons code for what amino acid. Here we have our first 2 codons from the mRNA sequence on the slide before. You can start off by finding the first letter of the codon, in this case A, the second letter U will come from the top, and G will be the last letter coming on the right. As you can see, this specific AUG codon codes for the amino acid methionine. Here, a mutation of G to C occurs in the DNA sequence. This is a point mutation as only one base was changed, and it also happens to be a transversion mutation as the purine base G was exchanged for the pyrimidine base C. The result is that the original codon GCT would have changed to GCU and then coded for the amino acid alanine. The mutation codon of CCT becomes CCU and then codes to proline. This effect is missense as the codon was changed and coded for a different amino acid, in this case alanine to proline. In our next mutation, T has become C in the mutant strand. This is an example of transition mutations as the pyrimidine T was changed to another pyrimidine C. The original codon TGA changes to UGA, which codes for a stop codon. However, the mutant codon of CGA codes arginine. This is a read-through mutation as a stop codon was changed to a codon that codes for an amino acid. In our third mutation, G is replaced with A. This is a transition mutation as both G and A are purines. The original DNA codon of TGG becomes UGG, which codes for tryptamine. The mutant strand codon of TAG becomes UAG, which signals a stop codon. This is an example of a nonsense mutation as the new base changed the existing codon into a stop codon. In our last example, T is replaced with A, representative of a transversion point mutation since T is a pyrimidine and A is a purine. The original codon GCT turns into GCU, which codes for alanine, whereas the mutant codon of GCA also codes for alanine. This is known as a silent mutation as the new base still codes for the amino acid as the original.